Okay, so thank you for the introduction. As said, my name is Anna-Carina, and unfortunately I cannot proceed with food or bread, but we will now go into a completely different topic. We go into life science, and to be specific, we go into the use of single-use plastics in life sciences. And here, my colleague Lena and our supervisor Peter, we worked basically on the future of single-use plastics in life science. Okay, let's go. So to show you the future for the single-use plastics in life sciences, we want to take you on a quick time travel. And we want to start with how life science labs developed in the past. And then I want to show you on how we think we can shape the future here. So as I said, even though here you can see future, we actually might start in the past. And to be exact, we start in the mid to early 1900s. And here you can see two famous scientists of their time. And what is more important here, you can see their laboratories and how they are equipped. Even though it's black and white, you might see there's a lot of glass, there is metal, there is even wood. And what the problem is here, that the cleaning is often not performed properly, and we have sterility issues. And then just a few years later, a very new and exciting material appeared on the surface, and it was plastics. And suddenly with plastics, everything seemed possible. We had single-use technology, we could produce a lot of products in a large quantity, very cheap, and life in general became much easier. So plastics and single-use technology also started a revolution in our life science labs. Suddenly we did not need to clean our equipment in an elaborate manner. New methods and new approaches were possible. Research became much more efficient. So life science, single-use plastics gives also a great opportunity for sterility. And why do we care about sterility? So when you think about a hospital, you think we need a sterile environment to protect the patient from harmful pathogens. So that's easy. Also in life science, we need to work sterile to ensure our product quality and again then patient safety. But as we talk about single-use plastics, you might know this movie. So if we continue living and researching in this big world of single-use plastics, we might become to this kind of reality at one point that plastic waste takes over the world. Toxic air pollution might make the earth uninhabitable. But, and stay with me here, if we see the potential in this technology, and if we can make a material that only draws the amazing opportunities and advantages that there is in single-use technology, we might be able to overcome this issue of plastic wastes and also the air pollution. So what do we want here? We want to have basically the best of both worlds. We want a material that does not require a lot of sterilization energy, and at the same time we want a material that does not accumulate a lot of plastic waste. So this seems to be an impossible task, right? It might be. However, to solve this problem, we need to be creative and we need to think, as you can see here, outside of the plastic Tupper box. And this is basically where our journey together began to find the future of single-use plastic in life sciences. So, as you can see, that's me, and we went far. And we thought 3D printing using bioplastics might be the solution. 3D printing is very cool. It can be very cheap. We can custom design it to fit our specific needs, and it would make us independent from supply chains, as we can print it in our labs. So still our main concern here is sterility. How can we make 3D printed bioplastics sterile to use it in the lab? I will tell you the solution when the slide changes. No, we found the solution. I will tell you right away. We found it exactly in-house. We found it in food technology, and that's amazing that two food technology presentations were before that. But we found it in the dairy industry. And now you might ask yourself, what does milk and single-use plastic in life science possibly can have in common? So it is the production of long-life milk. When we produce long-life milk, the milk passes an ultra-high temperature zone very quickly, which kills all the pathogens, but still leaves the milk healthy components intact. And we can use the same approach here for the 3D-printed lab equipment. So the 3D printing filaments pass the hot zone in the nozzle, and also here harmful pathogens are killed, which leaves us a sterile product. For our publication, we also compared it now to conventional single-use plastic in labs at the top, where you can see we can save a lot of energy in terms of transportation and manufacturing. And when you compare it to glassware, we can save energy in terms of sterilization energy. And as we all know, energy equals CO2 emission. So as an example, for our in-house sterilization, around 125 grams of CO2 equivalent are needed to sterilize one shake flask for one single cycle. And as we work a lot in our lab, this adds up really quickly. So we can save a lot of energy and therefore CO2. But now you might say, Anna, you talked a lot of plastic wastes. You didn't tell us anything about plastic wastes yet. So you are absolutely right. The question is, what about the plastic waste? And I will tell you, we also looked into that. So if we use recycled PLA, and if we recycle also the PLA we are used, then we can decrease the CO2 equivalent even further, and additionally, we can save a lot of plastic wastes. And this is exactly what we think the future of single-use plastics in life science looks like. It's the reduction of the CO2 equivalent by saving energy, it's carbon neutrality by using recycled bioplastics, and it's supply chain independence because we can 3D print easy all of our equipment in the lab. Yes, and this leaves me with our wishes for the future. And our wishes for the future are as easy as they are difficult. So we want to raise more awareness for sustainable research, and we want to start a rethinking of the production of single-use materials in life sciences. And ideally, on top, I would really like to finish my PhD soonish. And now the very last question is, why do we do that? Why do we even care? And the very, very simple answer is that we want to preserve the world for future generations. Thank you.